Hello everyone, we are solving question number 29 of K 2017. It says match the protein in group 1 with their cellular processes in group 2. So we have 4 protein P53, lysozyme, tubulin and histone in group 1 and we have to match these protein with their respective cellular processes. So let's begin with the first protein, that is P53. P53 protein is a tumor suppressor protein. It is a major effector protein in DNA damage activated kinase pathway. So whenever there is a DNA damage through radiation or any other factor, P53 gene get activated and it takes cell to program cell death, that is apoptosis. So P53 is linked with apoptosis. But whenever there is a mutant P53, which might be a case in many disease, P53 cannot perform its function, that is during the DNA damage, it cannot take cell to the apoptosis. So in that condition, it leads to uncontrolled cell division and ultimately oncogenesis. So P53 protein is related to apoptosis. So next protein is lysozyme. Lysozyme is abundant in human secretion including tears, saliva, milk and mucus. It is a part of first defense mechanism, that is innate immune system. It is an antimicrobial enzyme that leads to lysis of bacterial cell wall, and it is a glycoside hydrolase that catalyze the following process, that is hydrolysis of 1,4-B linkage between NAM and NAG. NAM is N-acetylmuramic acid and NAG is N-acetyl D-glycosamine, that is a residue of peptidoglycan. So peptidoglycan is the major component of gram-positive bacterial cell wall. So this hydrolysis leads to compromise of integrity of bacterial cell wall, causing the lysis of bacteria. So lysozymes are related to lysis of bacterial cell wall. So next protein is tubulin. Tubulin is a protein that polymerize into long chain or filament that form microtubule. Microtubules are the hollow fiber that serve as a skeletal system for living cells. So tubulin get polymerized. They first make alpha dimer of alpha tubulin and beta tubulin. These dimer get polymerized and make a long chain that leads to formation of macrotubule. So tubulins are linked with formation of macrotubules. So last protein is histone. Protein is the protein that provides structural support for a chromosome. So each chromosome contains a very long molecule of DNA and this DNA must fit inside the chromosome, as well as all the chromosome must fit inside the cell nucleus. So to do that, DNA wrap around the complex of histone protein, giving the chromosome a more compact shape. So histone proteins are linked with DNA packaging into chromosome. So let's match all the options. So we have P53, which is associated with apoptosis. Then we have lysozyme that leads to hydrolysis of polysaccharide and ultimately lysis of bacterial cell wall. Then we have tubulin that are important in chromosome segregation. How? Because they lead to formation of microtubules and thus these microtubules help in chromosomal segregation during cell division. And last protein is histone and they are involved in DNA packaging into chromosome. So answer is B.